So Dr. Gangloff is an associate professor of biology at Appalachian State University in Boone, North Carolina. He's been working with freshwater mussels in the Apalachicola Basin for more than 20 years. He first started working on the Apalachicola main stem in 2005. He's worked with and for the Army Corps of Engineers, U.S. Fish and Wildlife Service, the Florida Fish and Wildlife Conservation Commission, and most recently, he's been working with Apalachicola Riverkeeper on the project, our slough restoration projects, specifically on documenting the abundance and habitat of freshwater mussels in this unique system. One of the pictures that I had hoped to share with you before I stopped screen sharing, we did some sleuth towards the end of summer. The other important and fun thing to know about Dr. Gangloff, he's also a dad, and there had some fun pictures of his son in the field with us too. So he might share a little bit about that. But welcome, Mike, and we look forward to hearing from you. I will make you host so that you can screen share. Stand by.
Okay.
I think we're good. Thank you.
Working? Okay.
I see it.
Excellent. Well, there's the end of the talk. How about the beginning of the talk? Let's start there. Okay. Well, thanks, Georgia. Thanks for having me, and I appreciate everyone coming out today virtually to hear what I have to say about mussels. Just a kind of funny preface. I teach at Appalachian State, obviously, as Georgia mentioned, and I do a lot of Zoom lectures. But I was really not excited at the fact of having myself recorded and kind of begged to do a live lecture, because I kind of bragged to Georgia that I could stay on time and I wouldn't go over time and all that. And then I realized, of course, that I'd prepared a talk that was for a 45-minute talk instead of a 20-minute talk. So I'm going to go pretty fast and try to stay on time. But as I told Georgia, I'm used to people interrupting my lectures with questions, so feel free to jump in. I do have the chat kind of hidden, so if someone sees something pop up on the chat, just stop me. Otherwise, I'll keep yakking. Okay, so without further ado, so I consider myself an aquatic conservation biologist, really, and so we in my lab work on a variety of species, not just freshwater mussels, but the goals that we have kind of transcend species in aquatic systems. So if you were to kind of step back and talk about what the goals of conservation biology are, and this is basic information that my conservation biology classes get every semester. It's the first goal: document and describe Earth's biodiversity. The second is to understand the threats to that biodiversity, and then finally, and this is something that I think a lot of conservation biologists don't realize, is that even that we're our part of what we do is to also propose solutions to those threats to help mitigate threats to biodiversity. And so again, in my lab, we do all this sort of work, everything from genetic work to describe new species, to understanding broad scale climate and habitat change effects on species, as well as and working on projects that actually work to reverse some of these threats, habitat restoration, dam removal type projects. And those are really, in many ways, the most satisfying projects to work on, because you're actually making some positive change there. So although I do work on a lot of freshwater organisms, I'm most known for my work with freshwater mussels. So that's my nerd claim to fame. And that's something that a lot of people are kind of surprised to learn when they encounter me out in the stream. and they don't realize, a lot of people, that freshwater mussels are even down there. People who've lived on the rivers their whole lives generally do, but most of the general public that you're going to meet aren't very aware of freshwater mussels. And so the most common questions that my students and I typically get from the public include such awesome questions as, can you eat them? Are they zebra mussels? Which, short answer, no, generally we're not talking about zebra mussels here in the Southeast, but you can see in the lower right-hand panel, this poor elliptio from Lake Champlain that is completely covered up with zebra mussels. And then finally, of course, why should I care, or what good are they? And it's hard not to answer that with a cheeky answer. So I often have these kind of talking points for people who ask me these sorts of questions. So the first, well, there's really 5 things that make freshwater mussels super awesome from a biologist perspective. Here, especially in the southeastern United States, they're incredibly diverse, but North America is the global hotspot for freshwater mussel biodiversity. They have some pretty amazing life history strategies, incredibly long-lived animals, and I'm going to show you how they reproduce here in a second, which is mind-blowing for a lot of folks, including myself. They're important indicators of water quality, and that is in part due to the fact that they're long-lived species. Are also very sensitive to changes in habitat and water quality, and so those two things combined make them really good bioindicators. They're also ecologically very important. They fulfill a number of important roles in our ecosystems, and as we'll see, and this is the case with some of our Apalachicola mussels, quite a few are in pretty bad shape, highly imperiled. So why are mussels imperiled? Well, part of it is due to the unusual life history strategies. Freshwater mussels are fish parasites, and they have, in their larval stages, they have affected these really amazing adaptations to attract fish to them and infect them with their larvae. And if we have time afterwards, I could show you a couple of pictures of some of the lures that they've created, but really amazing. And so one of the things I like to tell people who seem like they might be a little hostile towards freshwater mussels is, hey, they invented fishing, and that's a pretty cool thing. They were doing that, hundreds of millions of years before humans showed up on the scene. I mentioned the sensitivity to disturbance, and the fact also that a lot of species, especially here in the southeastern United States, are highly endemic, makes them more susceptible to extinction or localized extinction. And finally, their habitats are highly fragmented places, right? And so this is just a map of the Mobile Basin in Alabama, Georgia, and Mississippi, a place that I've worked in for even longer than the ACF Basin. And this is just a map of the critical habitat units in the basin. And you might wonder, well, why the heck are there 25 different critical habitat units or 26 different critical habitat units in this basin? Well, it's because the entire basin is fragmented by a network of dams. I think there's something like 20 large dams in the Mobile Basin, and so that has led to an incredible amount of species loss, extinction, and endangerment. And the Mobile Basin has seen around 24 species of mussels and snails that have become extinct over the last 50 years, almost entirely due to the construction of dams and fragmentation of riverine habitats. Here in the Apalachicola River system, of course, we all are aware of its significance geographically as the largest river in Florida by volume. Its basin is also pretty massive and extends up into the Blue Ridge northeast of Atlanta. In much of the upper parts of the Apalachicola River Basin, there is no longer mussel habitat. Of course, the area around Metro Atlanta, most of those streams have been, completely destroyed by urbanization or water quality degradation. So really, when we're talking about mussel diversity in the Apalachicola Basin, we're talking about places like the lower Flint River, the lower Chipola, well, pretty much the whole Chipola River, and the main stem Apalachicola River. But even though we're talking about a relatively small proportion of this basin, those three rivers contain a pretty high proportion of the endemic fish, mussel, and snails that were found there historically. And though we do have 6 federally listed unionids at last count in the basin, there's been a relatively low number of extinctions in the basin. I think only 2 species in the Apalachicola Basin have gone extinct, which is, I guess you could say, better than the Mobile Basin. I, of course, don't think that any extinction is a good thing, but it has happened. So here's just a list of the species of mussels in the Apalachicola Basin. The ones highlighted in blue are our federally threatened and endangered species. And the ones where I've given you the common names, fat three-ridge, Chipola slab-shell, and purple bank climber, those are the species that I'm going to be talking about most during my my little talk here today, because those are the 3 endangered species that you find in the highest abundance in the lower Apalachicola and Chipola River systems. Just a picture of some of that diversity, and this image just shows that there's a tremendous range of shell morphologies. Some species like these anodontas are very thin shelled. You could easily crush them in your fingers, whereas others like washboards and elephant ears are very solid indeed, and you would certainly not want to get bonked in the head by one of those, because they can weigh 3 or 4 pounds and be pretty solid. And here in the middle, this is the fat three-ridge Amblyma neasleri, which is kind of our flagship mussel, if you will, for the Apalachicola system. So again, here are our 6 species. All 6 of these species do occur in the Apalachicola / lower Chipola system, but you have to go a little bit upstream of dead lakes to encounter some of these species. So over here, and they're in reverse clockwise order. So neasleri, Chipola slab shell, bank climber, shiny ray pocketbook, Gulf moccasin shell, and Pleurobiema piriformia, the Gulf pigtoe. You can find all 3 of these species in the middle and upper Chipola River, but they're not very common in the lower reaches of the Chipola. There's been a few sightings of Gulf pigtoe down there recently, but not a huge number. So again, Chipola slab shell, this picture's a little blurry, blurry picture of a beautiful place. Peacock Bridge, the Highway 278 crossing there. This is a threatened species that's endemic to the ACF Basin, like many of the threatened endangered species. And really the best populations are in this middle reach of the Chipola. You do find them though in the lower Chipola in pretty good numbers, and there have been a few in the Apalachicola. But really, if you wanted to go looking for these things, which we did when we were doing a genetic study of Elliptio, Peacock Bridge is one of the best places to encounter them. Our fat three-ridge mussel, again, this is an endangered species. It's only known from really the middle and lower Apalachicola and lower Chipola rivers. It never really extended very far up into the Piedmont, as far as we can tell from the historical data. And big sinuous river habitat here, like we see in this picture in the middle Apalachicola River is the place where you're going to find them in the greatest abundance. And then purple bank climber is a little bit more of an interesting species. This species is, I've actually made a mistake on my slide, is not an ACF endemic. It also occurs in the Ochlockonee, and it occurred prehistorically in as far east as the Swanee Basin. There are subfossil or Native American midden specimens of this species from that basin. So it's doing a little bit better than these endemics in terms of its range. It extends up into the Piedmont, and it likes rockier substrates than do the Chipola slab shell and fat three ridge. So you'll find it at places like Woodruff Shoals occurring in pretty good numbers in fairly deep water. And we think that is due in part to the fact that it's a sturgeon parasite and sturgeon spawn over these shoals. But it's also a large heavy shelled species. And so those, size selective sorting of stream particles extends to mussels, right? And so they're as many people have described them, rocks that suck. And so they often find themselves in the company of similarly sized rock particles. Let's see. Oh, questions in the chat. I'm sorry. Do we have any questions? Any momentum and dam removal on the southeast? Yes. Short answer. We could talk more about that later. Okay. As Georgia mentioned, I've been working in the lower Apalachicola, lower Chipola system since 2005. Before that, I had done a little bit of work in tributaries around Auburn, where I did my PhD in the headwaters of the Chattahoochee. But it really wasn't until 2005 when folks at EnviroScience hired me to help out with some surveys for the FWC that I really started working on the Apalachicola River system. And so I'm not going to talk too much on this slide because I've got some little vignette slides of each one of these talks or projects. So again, 2005, my buddy Greg Zimmerman, who works at EnviroScience, called me up and said, "Hey, we've got this project. I don't know anything about these mussels. I don't even have a permit to work down here, but they need us to come down right away and do these surveys." And this was a survey that was pretty comprehensive, included deep water work, but it was primarily focused on Swift Slough, which at the time was thought to be a really important freshwater mussel nursery habitat and was believed to have supported some of the largest populations of fat three ridge mussels in the system. Our research showed that while there were a fair number of mussels in Swift Slough, we also found other places in the system, including willow bar habitats in the middle of the Apalachicola River system and parts of the lower Chipola system that had high density fat three ridge populations as well. And in particular, one site, my job was just to identify mussels on the boat when the hard hat divers brought them up, and we were all pretty intimidated by working in this river. At least most of the biologists were. But hard hat divers who live at 500 feet in the Gulf of Mexico for weeks at a time call this cake work. So I was happy that I didn't have to do the diving, and they were out on the river one day doing some diving, and I said, "Just set me down on this sandbar," and I started mucking around in one of these little backwaters, and I realized that it was just packed with mussels, and it was packed with more fat three ridge at this one site than we had seen at any other site previously in the study. And so that got us thinking about the morphology of this bank habitat. And again, you can see in the background here, this willow growth starting to colonize the edge of the sandbar here. And that got us thinking, hey, maybe something's going on with these with these willow bars. And so Fish and Wildlife Service kind of took that ball and ran with it, and Karen Harrington and Sandy Percival really seized on these willow bars and the identification of what we call moderately depositional habitats as a way to look for additional places with high densities of fat three ridge mussels in the system. And so what they did in 2007 is they went out, they mapped all the willow bars in the river, and then when they were talking about relicensing or about flow level management at the time, they got the Army Corps of Engineers to fund a survey where we went to basically a subset of all these willow bars think we sampled 40 different sites along the entire river, and another 50 in the Chipola, or another 10 in the Chipola rather, for a total of 50 sites, to look at whether or not all these sites were occupied, and then to also assess the potential that drawing down the river to a level of 4,000 CFS, the minimum flow at the time was 5,000 CFS, what would the effect be on listed mussels? And so we used a hydraulic dredge and sieves to measure density. We wanted to be able to get estimates of all the little animals out there that you don't typically see, as well as the larger adults. And so here's Karen Harrington and me up in the top left-hand corner, and some of my students and colleagues, a number of whom work for the Fish and Wildlife Service now, doing some of the dredge work there. And you can see we've got a little keen gold dredge. I guess some people think it's fun to go out and dredge for gold in their spare time. I think it's a lot more fun to dredge for mussels, personally. What did we get out of that information? Well, we found that there were a lot more fat three ridge out there than really anyone had thought at the time. I think some of the population estimates that were coming out from the Army Corps of Engineers and others were suggesting, less than 100,000 animals in the river. And our estimate, just in those moderately depositional habitat, was that we were close to 750,000 animals, and probably a lot more. One of the things that we realized when we were doing this survey is that there were a lot more mussels further down into deeper water than where we were stopping. We were basically just doing that 1 meter depth contour. So that was pretty interesting, and would lead to our next study. We also saw lots of recruitment here. So these are baby fat three ridge mussels, and you can see they're itty bitty babies. So that led to the next phase of work, which was to figure out, okay, if mussels are not limited, or fat three ridge and other endangered mussels are not limited to these shallow water willow bar habitats, what's out there in deep water? Karen Harrington provided some of the initial seed funding to me to do some of the first deep water surveys in this time period, and we did find mussel beds in relatively deep water up to 24, 25 feet of depth. And so that got people really excited, and large numbers of mussels, because it suggested that mussel populations could be a lot larger than what we were thinking was actually there. So Adam Kaiser kind of took over the role of working on this species. Karen was, wound up moving to St. Louis around this time. And so Adam kind of stepped in, and he's a side scan sonar expert. That's his jam. And so he came up with the idea of mapping out all of the habitats in the river with side scan sonar, and then seeing if we could pick out mussel habitat using these side scan images. And so we did that over the course of, 3 or 4 years. He had a student working with him, Reuben Smith, out of Auburn, and we wound up coming up with a population estimate for fat three ridge that suggested this endangered species is actually the most abundant mussel in the river, and somewhere around 9 million animals is what we estimated in our recent paper that we published. And this is just a side scan image of the bottom of the Apalachicola River. These are in the middle section near Weewa, and you can see in the middle section these kind of dune-like ripples in the middle of the channel. And so that's, when you go down there, that's exactly what you see. It's essentially looks like the Sahara Desert, and so there's very little habitat in that middle channel section, the section that coincidentally is the area that had been dredged historically. But along these shallow bank, or along these bank habitats, IRZ stands for internal recirculation zone. You can read Reuben's thesis online if you want to get super nerdy on this. But basically, we could tell where the mussels were going to be by looking at these images. Wherever we saw these smooth bed forms in the river, we knew there were going to be mussels there, and our data really confirmed that. So this led to, well, not directly, but after that study, I got involved working with Dan Tonsmeyer and the folks out of Joanne Mosa and Matt out of Berkeley. Why am I drawing a blank on his name? The geomorphology guys. And we were looking at how Gangloff, that's his name. We were looking at how the river channel had changed over time, and we were interested in the dynamics of the sandbars in the river and how that dynamic had been shaped by historical dredge and disposal practices, as well as looking at possible projects for future restoration projects. And so one of the things that my job was to do is to look at how mussel populations had changed long term and try to identify, using some of these geomorphic data, some possible explanations for why those changes might be occurring. And this is one of the features that we were most interested in, these kind of, they were calling them hooks and bays, but these basically large sandbars that would protrude out into the river channel. And then you get this large area of slack water downstream. Note the willows starting to colonize this sandbar, so that's kind of nice to see. And sometimes these sand plugs would wash down and start to occlude the slough mouth. So here's a huge plug of sand near the mouth of Swift Slough. And that's, you know, one thing that we identified as a potential source of cutting off these sloughs. But from this study, I think the two most exciting take-home messages were, if you look at the top panel, that's just showing you fat three-ridge abundance over time. So just the raw number of animals found during surveys. You know, I've split this up into density and, you know, number per site, all that. And basically you still see the same trend with this really striking increase in the abundance of this endangered mussel beginning in around 2000. And granted, we don't have a lot of good data prior to 2005, but after 2005, we've got a lot of good data, and that's the most compelling part of that data set, in my humble opinion. Secondly, the lower panel shows that the places that had the highest number of mussels, and this is all species, tended to be the places where we had the most loss of open sandbar habitat, right? And so you might think, well, that's weird, that's eroding sandbars, that can't be good. But actually what we're seeing when we see sandbar habitat being lost, it's not that the sandbars are washing away, it's that they're being colonized by willows and eventually other floodplain vegetation. And so that is effectively stabilizing that sand and preventing it from moving. So those stable areas were the places that had, or the places that are stabilizing were the places that tended to have the most mussels. And again, you know, no matter how you kind of slice that data set, that trend was pretty evident. So that was pretty neat, and that's a data set I really need to get out here. But that kind of takes us to our current project involvement, and so this is a project that began in 2019, funded by National Fish and Wildlife Foundation via the Riverkeeper. And so in this project, we're looking at, and Ken told you all about the restoration of the Spiders, Douglas, and East River slough systems. And there, Ken, of course, told you all about the geomorphic and hydrologic modeling that they're doing. But my job is to do fish and mussel surveys pre and post restoration, so we can kind of see how these communities are responding to this restoration. And you know, this is a kind of a really satisfying, intuitively satisfying project because it gives you the opportunity to see how these restoration projects might work on a small scale, and then how they might be applied at larger scales. We kind of have three different scales of restoration that we're talking about here, with some of these sloughs getting a little bit of dredging and others potentially a lot more restoration work. So back in last August, we did some scouting, and there's me and my son Wade he's starting the initial dredging of the mouth of Spider Slough there. I think that's something that we all, maybe even before we're paleontologists, we start off as engineers digging in the sand. So we were able to get out in November and do a little bit of mussel and fish sampling, but conditions were kind of in that range where they were a little too high. The water was a little too high for effective mussel sampling and a little too low for effective fish shocking. So we're hoping to get back out there this spring and wrap that work up. We need to do a little bit more work in East River in particular. But what we found so far is that there are Fat 3 ridge mussels in both the Spider Slough and Douglas Slough systems. East River appears to be a little less diverse, and you start to see more salinity tolerant species. I'm not sure. There are very few records of mussels in general when you get below Brickyard Island on the river, and I'm not sure if that's due to the fact that there isn't a lot of mussel habitat, or it's because that part of the river is pretty scary. My crew pretty much mutinied on me after we had like 5 gators jump into the water with us on East River back in November. So we'll have to bring the gator repellent next time we go. But we didn't see any other listed mussels down there, but we're gonna have to get back in there and do a little bit more work before I'm satisfied with that conclusion. And just some pictures from our November exploits. Here's Douglas Slough, and we found Fat 3 Ridge at this site. The water was fairly clear in Douglas, not a lot of movement. Here we are in the crew in East River, talking about whether or not they're more likely to get COVID from each other standing so close, or eaten by an alligator. And there's some glebula down there, a mussel that can tolerate salinities of up to 12 parts per thousand. So that's kind of what we expected to see down there. And again, no Fat 3 Ridge or any other listed mussels to date. And here's the fish crew behaving themselves, doing some socially distanced sampling with masks on in the jungle. So we had good luck up at the headwaters of these streams, but the lower ends of the sloughs, we need some different equipment to effectively sample these. When it's a foot deep, you can get in there with the seine, but not so much when it's 10 feet. Anyway, so that's what I have for you today. Stay tuned for more updates from the sloughs, but I'd like to thank everyone for their attention, Georgia for inviting me, and a range of folks, agencies, and other entities for funding my work, as well as all the people who put life and limb on the line to study the mussels of the Apalachicola River. And fortunately, the only places where the gators on the Apalachicola are very friendly is up at Woodruff Dam, where they're fed. All the other ones mostly left us alone. So with that, some pictures of the dive crews that have worked out there, and you can see you've got to be a little bit crazy to do this, and it also helps to be 25 years old. So I rely pretty extensively on my graduate students and folks like that. But anyway, thanks for your attention, and with that, I will take any questions.
I'll lead the round of virtual clapping. Thank you, Dr. Gangloff. That was really fabulous. Katie, could you facilitate questions? I'm wrestling dogs. For those of you that don't know, I have 3 big unmannered dogs.
Yes, I'd be happy to. And I do see we do have some questions in chat. One from Josh Fitzhugh. Are there any saltwater mussels near or in the bay? If no, why?
Well, so the animals that I primarily work on don't like salt water very much. They're very freshwater, and they're very different from, evolutionarily speaking, from the mussels that we would find in brackish water or marine habitats. So there's kind of this transition that occurs as you get down into more estuarine and tidally influenced reaches, and you can see it just by going across the river, actually, because the salt wedge moves up the river, and so the deeper parts of the river, I would expect down around the delta there to have mostly brackish water estuarine species, whereas the freshwater species would be restricted to the edges of the river where you have kind of a freshwater lens. So there's a transition that occurs there.
Great. I see another question from Kim Sash about how is the water clarity?
It's variable. It's variable. It's so yeah, the river in September, October, November, when the flows are at below 8,000 or 9,000, the river is pretty clear. The Apalachicola is pretty clear. The Chipola really never gets clear for whatever reason. It tends to have a lot more suspended sediment. So you're talking about like maybe 1 to 3 feet in the Chipola, and on a good day, 6 to 8 feet in the Apalachicola. Not bad.
Thank you. Craig asks, any thoughts on optimal slough redesign in that project to bolster populations?
Well, I think that the big thing, as far as mussels are concerned, with the sloughs, is how much connectivity is there going to be to the main stem river, and how much water is going to stay up in those sloughs when the flows get low. When we were there in November, we thought conditions were pretty low. Like, I mean, we could work most of the sloughs without having scuba. We were able to just snorkel, or death snorkel we call it, because we wear a weight belt and a mask and snorkel. So it's a little bit sketchy, but for 5-6 feet of water, that's a pretty good way to go. But when we were there in August, when flows were they were around 9,000, there was very little water in those sloughs, and so we were seeing large stretches of them immersed, dried up. And so when the sloughs dry up, then the mussels are not going to survive for very long. So that's I think the main issue there is just improving connectivity. And in terms of like the morphology of the slough and the sinuosity, I would leave that to I'll leave that to the geomorphologists. But what mussels are just looking for is a little water up in there.
Excellent, thank you. And I see one more question from Donna Wells. What is the choice eating type mussels, and what is your favorite way to eat them?
I really just like mussels and a little butter with garlic and a nice nice Belgian beer, something like that. That's a really good way to eat bank mussels. But I would not recommend eating freshwater mussels, although of course Native Americans ate them pretty across a pretty broad area and for quite a long period of time. And you can actually see Aboriginal middens eroding out into the Apalachicola River in places. There's one just downstream of Bluntstown that's full of it's actually full of extinct mussels. It's kind of cool, among other things. But yeah, so but I think that was probably a response to like crop failures or some other sorts of cataclysms, because most people do not think that freshwater mussels taste very good. I have not tried them.
Thank you for that. And a big thanks, Dr. Gangloff, for your presentation. Hand claps for everyone who has video on. We very much appreciate it. And
Sure.
thank you,